Today, I would like to present our hackathon project, Project Alpha. Start small, dream big. So we've come up with an idea of AI-powered immune rejection discovery platform. The problem, immune rejection occurs when a transplanted stem cell is not accepted by the body of the transplant recipients. Basically, when a new organism enters the body of a human, the immune system will examine its genes and determine if it is from the body or from somewhere else. And if it is a non-cell gene, the immune system will react and fight back. Stem transplant is the transplantation of human cells that are able to develop into many different cell types. A famous example would be embryonic stem cells. 60 to 75% of kidney recipients and 50 to 60% of liver recipients will experience acute rejection. Meet Jared. Jared is a 13-year-old who was diagnosed with leukemia 5 years ago. He received chemotherapy, enabling him to graduate elementary school. However, 5 years later, leukemia came back. Doctor infused him with a bone marrow transplant from his brother, but once again, leukemia returned. What he's experiencing is his immune system fighting it back because of a non-self cell in his body. Current solutions that are trying to tackle this immune rejection problem. So fail-safe system from Pensela is by adding a suicide gene to transplanted cells, it will eliminate the transplant cell if the immune system overly reacts, therefore causing no harm to the body if this transplant cell doesn't work out. The other company called Blue Rock Therapeutics wants to develop a universal stem cell which is capable of unlimited expansion into any cell type in the body. What we are trying to present is a solution that complements with current solutions. What we've discovered through research are the two main causes of human immune system rejection: human leukocyte antigen system mismatch or minor histocompatibility complex mismatch. HLA are self-surface proteins that are responsible for the regulation of the immune system, encoded by the major histocompatibility complex. HLA have thousands of variant alleles under different classes, which are different forms of a given gene variants. So when you're trying to transplant the stem cells, you want to match as much HLA as possible between the donor and the recipient in order to not have immune system rejection. For example, here we can see that the donor is donating a stem cell with HLA A, B, C. And we can see that the HLA B and HLA C from the donor is entirely different from the HLA B and C from the recipient. Therefore, the immune system will identify this as a non-self HLA gene, which will trigger the immune system to fight back this allo antigen. What we want to do is, through the recorded reaction of the HLA alleles, use this data to train the ML model, which can then predict future alloimmunity of different alleles combination. In this example, the raw data would be the HLA A, B, and C from the recipient and donor as training data set, and the desired outcome would be the results that the doctor has recorded when he decided to follow through with the treatment. And through algorithm and processing, we can predict future cases where the immune system will fight back and the degree of how it will fight back on the other combinations of HLA from the donors and recipient. As stem cells treatment becomes more common, our AI model will act as a testing medium for potential treatment. Therefore, no need for more human testing and trial and error. We can do this by using AI.